Today we will be going through the polymerase chain reaction for the EdXLB A-Level Biology syllabus. The polymerase chain reaction is a method used by many scientists and forensic experts to amplify DNA samples into millions of copies so they can be analysed and used for many purposes. In this video, we will cover the structure of DNA, the method of PCR, how PCR is used in practice, and finally, we will show you a typical exam question used to test knowledge. To understand PCR, it helps to have a good understanding of the structure of DNA, so here is a quick overview. DNA is a double helix structure consisting of millions of base pairs. Each DNA strand is made up of a pentose sugar, a nitrogenous base, a phosphate group. These three parts join together to form a nucleotide, and millions of these all join together with hydrogen bonds, forming the sugar and phosphate backbone of DNA. Now we will go on to explain the process of PCR. To carry out PCR, you need a PCR machine, the DNA fragments that you want to amplify, primers, which are 4 base pairs long, 3 nucleotides, DNA polymerase, and a buffer to maintain the pH of the reaction mixture. These components are all mixed in the PCR machine for the process to begin. The mixture is heated to 95 degrees in order to break the hydrogen bonds between the DNA nucleotides. This then separates the 2 DNA strands. Separating the strands then allows the next step of PCR to occur. The mixture is then cooled to 55 degrees. The DNA polymerase needs primers to be able to bind to the DNA strand, as it can only bind to double-stranded sequences. This process occurs simultaneously on the 2 strands separated in the previous step. The mixture is then reheated to 72 degrees. Once bound to the end of the DNA fragment, the DNA polymerase is able to move along the strand, adding the 3 nucleotides from the reaction mixture. This creates new double-stranded DNA fragments identical to the original fragment. The process of PCR is repeated thousands of times, each time doubling the DNA content of the mixture. This is known as exponential growth. We will now explain how PCR and its products can be used. PCR has many uses. One of these is analysing DNA found at crime scenes. At a crime scene, there may only be trace amounts of DNA. A sample of this can be collected and PCR used to amplify it. This creates sufficient copies of the DNA so that it can be analysed by different forensic scientists, allowing identification of suspects. PCR can also be used to diagnose viral infections. The DNA in the cells is amplified and is then compared to reference viral DNA samples on a database. This allows identification as to which virus an individual may have contracted. Here is an example exam question about the polymerase chain reaction. It is worth 4 marks, so should take about 5 minutes to complete. Pause the video and come back once you've had a go, and we'll go through the mark scheme. Now let's have a look at the mark scheme for this question. First, you should say that you will use PCR to amplify the sample. You then get 1 mark for mentioning each of the following up to a maximum of 4 marks. So one of the points you could have said is adding DNA polymerase to the mixture. Another point is using primers and 3 nucleotides. Next, you could have mentioned any of the temperatures used throughout the process, but they must be within the ranges stated on the mark scheme. And finally, you should mention that the cycle is repeated several times to get multiple copies of the DNA. That is all you need to know about PCR. We hope you found this video helpful, and thank you for watching.